Hi everybody, and welcome to our skeletal system presentation, skeletal muscle system presentation, part 2. We are going to be focusing on muscle contraction for this part. Now, when you guys look back at what you learned for the nervous system, more specifically the somatic branch of the nervous system, we learned that it controls the voluntary movement of the muscles, which implies they control the skeletal muscle movement. So, as it says, the skeletal muscle contraction requires input from your nervous system. Now, nervous system cells are neurons, which we feel very comfortable with since we have discussed it in detail, or nerve cells. And the way that your muscle contraction happens is neurons communicating and passing their electrical impulse to your skeletal muscles, which again causes muscle contraction. So I'm not going to spend a significant amount of time on this. This is more of a review rather than you guys starting and getting to know the structure of the neuron from the beginning. So we know neurons create electrical impulses or action potential. Well, they actually create graded potential and action potential. But these electrical impulses start from the dendrites, pass through the cell body, and down the axon and end at axon terminal. Axon terminal is the location where you store all of your neurotransmitter. In this case, for the skeletal muscle contraction, the only neurotransmitter you will be dealing with is ACH or acetylcholine, which again should sound very familiar from our previous lectures. So here is the depiction of how a neuron communicates with a skeletal muscle. So you have your axon, you have your axon terminal. These vesicles represent your vesicles carrying your neurotransmitter ACH, or again acetylcholine. These basically neurotransmitter gets released into the gap between the skeletal muscle and the neuron. The gap is still called the synaptic cleft, exactly like we discussed in terms of nervous system. The only new thing that is present here is the type of channels that are specifically present in the skeletal muscle. So in the skeletal muscle, the only channels we have in terms of the neuron to muscle communication is sodium-potassium channel. And again, this should sound really familiar since we have discussed it in a lot more detail in terms of nervous system. But let's again kind of review this concept. First, the area where the nervous system, or more specifically your nerve cell or your neuron, communicates with your skeletal muscle, that area of communication, which is right here, is referred to as the neuromuscular junction. So this is similar to the synapse when two neurons communicate. But in this case, because it's neuron to muscle, we refer to it as the neuromuscular junction. Again, the area between that where the neurotransmitter is released is referred to as the synaptic cleft, like it was in terms of neuron communications. The only, again, point here is that the only neurotransmitter involved in this process is your ACH. So looking at the steps, electrical impulses in the neuron passed on all the way to the axon terminal, leads to creation of electrical impulses in your skeletal muscle cell, and that eventually leads to muscle contraction. So how does that take place? So we're going to go into a step-by-step picture, kind of giving you an idea of what you already partially know, but again, kind of going into a little more detail. So I'm going to use the picture to walk you through the steps, and then we'll go back to the step-by-step description on the previous slide. So here is my axon. Axon ends at the axon terminal. Now at the axon terminal, what we have is we have these calcium voltage gated channels. So it's here it just says calcium, but remember these are calcium voltage gated channels. So electrical impulse or the action potential that is coming down, which is these purple arrows, causes the calcium VGCs to open. And when these channels open, calcium is going to influx into my axon terminal. Now, the influx of calcium causes the vesicles containing ACH to move toward the end of the axon terminal and release their material into the synaptic cleft. Now, this process that is depicted, the release of your ACH into the synaptic cleft is through process of exocytosis. It should sound familiar to you. Now, what you have is that under normal condition, on the sarcolemma of my muscle, we have these channels called sodium-potassium channels. And in the absence of my ACH neurotransmitter, these channels are closed. They're not active. No ions moving in or out of my muscle. However, as soon as the ACH gets released into that space, that causes the that causes the ACH to bind to my sodium-potassium channel and open up those channels. So as soon as my sodium-potassium channel open up, that causes the sodium to influx, sodium influx, remember double letter influx, and potassium, my single letter efflux at the same time. Now here's the important part. Influx of the sodium happens at the faster rate compared to the efflux of potassium. In another sense, you're gaining more positive charge than you're losing positive charge. So that gain of positive charge basically causes depolarization of the membrane within the neuromuscular junction. So kind of going over what we learned. Nerve impulse travels down the axon and reaches your axon terminal, right? At the axon terminal, we have your calcium voltage gated channels. Calcium comes into the axon terminal and causes the exocytosis of ACH into the synaptic cleft. Basically what we depicted right here, right? So ACH gets released into the synaptic cleft. Now, ACH binds to your sodium-potassium channels that are present at the sarcolemma. It causes sodium influx to happen at a faster rate or higher rate compared to your potassium, and that leads to the depolarization. Now, here's the new steps. The depolarization of the depolarization wave that is now inside my muscle fiber needs to spread farther throughout my cell. And for that, it uses the T-tubules. Now, T-tubules, again, remember, are the extensions that dip down into my muscle fiber. So basically, that's what they are going to allow the depolarization to spread. You also learned that the T-tubules were surrounded by sarcoplasmic reticulum and terminal cisternae, which contain a significant amount of calcium. Now, these calcium are stuck inside the sarcoplasmic reticulum, and their opening to the outside is protected by calcium VGCs. So when T-tubules spread the electrical impulse, it causes that calcium to be released into your sarcoplasm, which is the space inside the muscle cell. And as we talked about earlier, calcium can interact, going back to what we talked about earlier, calcium can interact with what? Calcium can interact with my troponin and move tropomyosin, which allows the myosin binding site to interact with the myosin heads, and that interaction causes muscle contraction. So yes, I know it's a lot of information, guys. So what I'm going to do, kind of showing you this process in a kind of a simplified version. You have, sorry, I'm going to go back. Right here, you have, again, sorry, give me 1 second. So I'm going to kind of depict this for you guys in a really simple version of the same picture. So what we have is, again, here is my axon and axon terminal. Here is my, try to say it with me as I'm writing it, guys. This is my synaptic cleft, right? These are my vesicles with ACH, right? This space is my axon terminal. This is my axon. This membrane here is my sarcolemma, which is my plasmamembrane. Right on this side, I will have what type of channels, if you guys recall? These are sodium potassium channels, right? That are sensitive to my ligand ACH. We also have channels right here. And these channels right here are going to be calcium voltage gated channels, right? This continuation of the plasma membrane that is dipping inside, that's my T-tubules, which is surrounded by my, and again, this is a simplified version, surrounded by the sarcoplasmic reticulum. And inside you have a lot of calcium. Now this calcium cannot be released until you have your little calcium VGCs open. So these are my calcium VGC. So looking at this picture, electrical impulse comes down, ACH gets released into the synaptic cleft by relying on the calcium VGCs. ACH comes and binds to the sodium potassium channels. That creates the initial depolarization wave, which causes eventually, and I'm gonna use the word eventually, for the electrical impulse to travel down through the T-tubules, which is going to go and causes the calcium VGCs to open. Calcium leaves and comes into this space, which is your sarcoplasm. And now inside the sarcoplasm, you have your actin and myosin. So look, here is my myosin. And these are my myosin heads. And then you have your little actin, right? And the way these actin and myosin are interacting is the calcium coming in. I'm going to use a different color. And moving, kind of moving these troponin. So this is tropomyosin. Tropomyosin and troponin out of the way. And again, calcium comes in and interacts with that. Calcium comes out, interacts with the troponin, which interacts with tropomyosin, and allowing these to interact, and that causes the muscle contraction. It's a very fascinating process, guys, and understand every time you move your muscle the smallest amount, every time your muscle contracts, even if you do the same motion 10 times, 20 times, 100 times, this process happens over and over again. Now, here's a thing that needs to be clear to you guys. So as of right now, what I've told you is that I have ACH coming and interacting with the sodium potassium channels, and that causes basically the depolarization, which eventually travels down the T-tubules. So what is happening from here, from sodium potassium channels, which are localized within the space to all the way to the T-tubules? How do I transfer the electrical impulse in this distance? So for that, we are relying on, what do you know? Your sodium channels and potassium channels. These are same voltage gated channels that we described in terms of your neurons. So when sodium VGCs open up, they cause the sodium to rush in, and that creates my action potential. And then by the time my positive charge is done, you have depolarized your cell. Sodium VGC is going to go into inactive, and my potassium VGCs are going to open, causing the potassium to leak out, and that again causes the muscle to repolarize. So here is what I'm telling you guys basically. You have the initial opening of the sodium channels that are your sodium potassium channels, and that causes the fast opening of your sodium voltage gated channels, causing this again really big positive rush that you see right here. And then at this time, your sodium channels are going into closed or inactive, and then you are repolarizing your cell down through using your potassium efflux or potassium exiting your cell. Now, by the time you reach back to the original voltage, your potassium channels are also closed, and same as before, same as we described before, you're gonna use your sodium potassium ATPase to return your ions back to their normal place, as a sodium outside the cell and potassium inside the cell. This is the last picture I'm going to talk to you guys in this PowerPoint, and then we'll continue the last part of this presentation on part 3. So looking at some of the structures that you guys need to know here is, again, kind of highlighting the same points: axon, axon terminal, ACH being released, electrical impulses created and then traveling down the T-tubules. And that causes the calcium to be released from your sarcoplasmic reticulum, which is what you see here, zoom in version. So electricity comes down, sodium VGCs open up, potassium, I'm going to say every ion now. Calcium VGCs are going to open up, and calcium is going to basically get dumped, and I really mean dumped. It's a significant amount of calcium dumped inside the muscle fiber or within the sarcoplasm. Now, notice again, these calciums are going to come and bind to the yellow parts, which were my troponin, which causes the troponin to move tropomyosin, which is the line, the little orange line. And now you have these little opening on my myosin binding site of the actin that can interact with the myosin heads, and that creates basically what we identify as a cross bridge, which causes my muscle to contract. So there are quite a few questions, guys, in terms of understanding the picture and identifying parts of it, and then also identifying the process that is involved, starting with electrical impulses traveling down the axon all the way to the actual force that, actual interaction between myosin and actin that causes the muscle contraction. Okay, so we're going to stop here and we'll continue for our next PowerPoint later on.